The first question says, which statement about light microscopes are correct? To calculate the magnification of a light microscope, the half-pitch lens and the objective lens magnification are added together? No, they are not added, but you find their products, they're multiplied, not added. So that's not correct. The next one says, the resolution of a light microscope is limited by the length, by the wavelength of light. That's true. We all know that the light microscope has a low resolution because of the high wavelength of light, the relatively higher wavelength of light when compared to an electron microscope, which is an electron with relatively lower wavelength of light. The division on the scale on the stage micrometer are closer together. That's not correct. Than the division on the scale of high-pitched particle, of course, like I said, it's not correct. So the only correct one here is 2 only. 2 only. That's option C. The student measured the width of a mitochondrion in an electron microscope of an animal cell with a magnification of times 9,600 and the width was 6 millimeter. What was the actual width of the mitochondrion? So the formula is magnification equals image size over actual size. Magnification is 9,600. The image size is 6 millimeter. The actual size is what we're looking for. So we'll bring this here, take this close here. That's A equals to 6 millimeter divided by 9,600. When you do that, you have 6 divided by 9,600. That gives 0.000625. And in micrometer, you multiply by 1,000. You have 1, 2, 3, which is 0.625 micrometer. And to 1 decimal place, you have 0.6 micrometer. The photomicrograph shows a section of the stem of a plant. Okay, so this is the transverse section of the stem of a plant. And of course, this is showing out the vascular bundle, where you see the xylem. And remember that xylem has P, which was originally plasmodesmata in the young xylem. When it became mature, that's part of the plasmodesmata that was not dignified becomes peat. So the cell wall contains peat. That's correct. Cell wall contains cellulose and also lignin. That's correct too. But it's connected to adjacent cell via plasmid matter. That's not correct. So the answer to this question is 1 and 2. The diagram is taken from an electron microscope, micrograph. And here we're simply asked of a cell that secretes enzymes. We're simply asked where the polypeptides, chains of amino acids of the enzymes are made. They're either made in ribosomes or rough endoplasmic reticulum. And here is the nucleus, the mitochondrion, and here is the rough endoplasmic reticulum, the Golgi body, the secretory vesicle. So this is the answer. The rough endoplasmic reticulum is the answer. Which comparison between a typical bacterial cell and a typical plant cell are correct? Cytoplasmic DNA, nuclear DNA. That seems to be the correct one. That's correct, right? No smooth ER present. Smooth ER present. That's also correct. Peptidoglycan cell was found. Cellulose cell was found. Also correct. This is supposed to be the other way around. 88 ribosomes for plant cell, 78 ribosomes. So the only correct ones are 1, 2, 3. This option A. To the next one, number 6. The diagram shows the virus. This virus looks similar to the HIV virus with a phospholipid envelope. Phospholipid envelope. This is a phospholipid envelope. This is a capsid made of protein. The capsid is made of protein. Then this is a nucleic acid. So here, the nucleic acid is either a DNA or an RNA. It cannot be both. So X is phospholipid. It has to be either this or this. Y is protein. W is DNA or RNA. So, and Z is a glycoprotein, like a GP, glycoprotein. The iodine test and the Benedict test were carried out on samples of a starch solution. Amylase was added to another sample of the starch solution and it could be there for 10 minutes. The Benedict test was then carried out on this sample. So basically here, iodine test before adding amylase, there will be starch. Before adding amylase, amylase breaks down starch into maltose, which is a radiation sugar. Maltose is a radiation sugar. So before adding amylase, if you carry out iodine test, you're going to have a positive result. But when you add Benedict test before adding amylase, because it's only starch you have there, and starch is not a radiation sugar, you're going to have a negative result. So this looks like the answer. This is looking like the answer already. You can see it's beginning to look like the answer. So Benedict test after adding amylase. Of course, Benedict test after adding amylase will be positive because amylase would have broken down starch into maltose, which is a reducing sugar, and gives a positive test with Benedict solution. So the answer to this question is C. We go to number 8. Which rule about alpha glucose and beta glucose molecule is correct? Remember, when you have these two molecules, the difference is at carbon 1, the OH, carbon 1, here the OH points upwards for beta glucose and points downward for alpha glucose. And this is found in cellulose, allowing cellulose to form the rigid or strong structure that it needs to provide support in the cell wall. And this is found in the storage polysaccharides like glycogen and starch, allowing them, of course, to store up a lot of glucose for energy. So carbon atom on which the position is different is 1. Cellulose contains both molecules. Cellulose only contains beta, so cellulose contains both molecules. That's no. Which molecules are monosaccharides? Of course, when we talk about monosaccharides, we have 3 carbon, we have monosaccharides are simple sugars, and it can be 3 carbon, 4 carbon, 5 carbon, 6 carbon, like that. So this is a monosaccharide, 5 carbon monosaccharide, 6 carbon monosaccharide, a 5 carbon monosaccharide, but this is a disaccharide. So the answer to this question is 1, 2, and 3. Now go to the next one, number 10. Many animals use triglycerides for long-term energy storage. Yeah, because they can store energy longer for us. Which statements are correct? Additional functions of triglycerides, apart from being long-term energy storage, yes, they also provide buoyancy in some marine animals like whales have what we call blubber, right? Yeah, it's a fatty substance that gives them buoyancy in water. Also, main component of cell membrane, that is not correct. The main component of the cell membrane should be phospholipids and not triglycerides. Triglycerides are mainly for storage of energy. Phospholipids are the ones found in the membrane. Triglycerides do not have the structure that would enable them to function as cell membrane structure, or as cell membrane component rather. So this is out of it. They are used as thermal insulation, yes, in our skin, where they help to, under our skin where they are stored. They can help prevent loss of heat, especially when it's cold. So we have 1 and 3 only. We go to the next one. The next one says, the diagram shows 3 different bonds. Yeah, this is a bond between hydrogen and a more electronegative element, oxygen. This is, of course, this is hydrogen bond. This is a bond between a carbonyl and an amino group. Of course, this is an amide bond, the way we have it, or peptide bond, the way we have it in the primary structure of proteins. And this is a disulfide bond or linkage. So which bonds are found in proteins? This is found in the primary, this is found in the secondary, tertiary, and quaternary structure. This is found in the tertiary and quaternary structure of proteins. So they are all found in proteins. What describes the quaternary structure of collagen? Of course, when we talk about quaternary structure involves the interaction between 2 or more polypeptides. So a protein that has 2 or more polypeptide subunits would have quaternary structure. But a protein that has only 1 polypeptide, like myoglobin, would not have a quaternary structure. The highest structure will be a tertiary structure. But for collagen that is made up of 3 polypeptides, like we know from our coursework, 3 polypeptides, They form triple helix. So three polypeptides that form helix structures, triple helix, which are held together by hydrogen and covalent bond. Not alpha helix, not double helix, but triple helix, like three intertwined ropes. The diagram shows the structure of amylase, and we are shown a three-dimensional structure. This is a 3D structure of how it is folded. When I talk about the 3D structure of how it's folded, regular and precise folding of the protein. That's a 3D structure, and that's the tertiary structure. That talks about the tertiary structure of the protein. Which statement explains why large volumes of water maintain a constant temperature? That's because the temperature of water does not rise easily due to the high specific heat capacity of water. The amount of heat that is needed to raise the temperature of water by 1 degree Celsius, the temperature of 1 gram of water by is the specific heat capacity, and it's high. So the temperature of water does not rise easily. That way, water is able to maintain a constant temperature even when air temperature increases. This statement about the mode of action of an enzyme are correct. Some enzymes have a region on their surface to which substrate has a complementary shape. Okay, so that's the active site. Enzymes and their substrate can change shape slightly as the substrate enters the active site. This is the induced fit model. It's talking about the induced fit model. Enzymes permanently change shape. Enzymes do not permanently change shape when they react with their substrate to form. No, they do not permanently change shape. They release and the active site goes back to normal so that they can bind substrate again. Here we have the graph showing us the effect of increasing substrate concentration on the rate of reaction of two enzymes that catalyze reactions with the same substrate. So here we have the curve for the two reactions. And this statement is talking about Km, right? And when you're talking about Km, Km is the substrate concentration at half Vmax. So locate the Vmax. To locate the Vmax for this particular reaction, we are locating something around here. You're locating the Vmax here, and it's just here. And the half Vmax for this one will be around here. So substrate concentration is going to be at half Vmax will be around here. Or if you look for the substrate concentration at half Vmax here, it's going to be very much closer around here. So this one has the lower substrate concentration at half Vmax, that's enzyme Q, which is Km. That's the Km. So the Km, which is substrate concentration at half Vmax, is lower for Q. And the lower this substrate concentration, which is the Km, the lower the Km, the higher the affinity of the enzyme for the substrate. So enzyme Q has a lower Km and the higher affinity for the substrate than enzyme P.
A lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Ajiro Tech do have a private tuition. So by my right, you can see that we have for the EdExcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have um, you know the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, I think all you have to do is send an email to the sales team at ajirotech@gmail.com, and they would reply to you, talk about your time zone, talk about, and the fee is really quite affordable. Really, it's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable and you get the very best of service. We also have for the E levels as well, and also for the edXL. So we have for the Cambridge E levels and edXL E levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E level, mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time, even physics, for E level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid, okay, and send an email to the sales team at agile@gmail.com and they will reply to you.
The cell surface membrane structure is defined as fluid mosaic. Fluid because of the random motion of the phospholipid in the bilayer. Mosaic because of the pattern, the way the proteins are scattered within the phospholipid bilayer to give a mosaic pattern. It's not a regular pattern, and it's not produced by phospholipid L, it's produced by the proteins. The proteins are scattered within the phospholipid bilayer. Question 18. A student immersed a plant tissue in solution X. For the next 30 minutes, the students used a microscope to observe the cells in the plant tissue. During this time, the cytoplasm of the cell shrunk and no longer pushed up against the cell wall. In many cells, a space appeared between the cytoplasm and the cell wall. A space between the cytoplasm and the cell wall. So this is the cell wall. Then we have the cytoplasm pulling away from the cell wall. Which statements are correct conclusions from these observations? The water potential in solution X was more negative than the water potential in the cytoplasm of the cells at the start of the experiment. Of course, yes. That's why water had to move out of the cell. Now, without a cell wall to provide support, it is likely that many of the cells would have burst. Of course, this would have been the case if water moved into the cell. That is, the water potential was of the solution was actually less negative. But in this case, it is not. This is not correct. The clear space between the cell wall and the cytoplasm of many of the cells was filled with air. We're told that is a clear space from the question, a space. Okay, so that also crosses it out and makes the answer to the question option B. Now, number 19. Also, according to this question, the question here, number 19, A sample of 500 cells dividing by mitosis was examined to identify the stage of mitosis for each cell. And the table shows the results. Okay, now we have late prophase, end of metaphase, then between the start of anaphase and the end of telophase. Now, which role is correct? The number of cells with chromosomes at the equator of the cell. Of course, that will be at metaphase, cells at the equator, metaphase. And that brings us to 45 over 500 times 100 over 1, which gives us 225. 225. Then we have number of cells with separated chromatids. We can only see number of cells with separated chromatids at anaphase. At anaphase and the end of telophase. So by the start of anaphase, the chromatids start moving to the opposite they start moving to the chromatids start moving to the opposite end of the, so they are separated. So that would be 24 over 500 times 100 over 1. And that gives us 120, which makes A the correct answer. The diagram shows a chromosome. Of course, we know this part is called the centromere, holding the two chromatids, sister chromatids together. And this is the telomere that prevent the loss of vital genes. So centriole is not correct. Centriole is a subcellular structure that forms microtubules during mitosis. The centromere and telomere. Now for number 21, which processes are used by stem cells during tissue repair? Of course, stem cells will divide by mitosis, and afterwards, the cytoplasm will divide into two to give rise to more cells that will repair the tissue. And before division, there is DNA replication. Right. Also, when we look at transcription, transcription has to do with the cell passing through transcription has to do with a stage in the cell cycle. Transcription is it's actually a part of G1. Yeah, it's a part of G1. And there is the cell goes through, there's M phase, there's M, there's G1, there's S, and there's G2. So if S is part of S, which is DNA replication that occurs there, and G1, during G1, the cell carries out its normal function, which includes transcription of genes, translation of mRNA into protein. So the three processes are involved. Now the enzyme telomerase, what is the role of the telomerase? The telomerase is always topping up the telomeres, keeps adding to it so that it doesn't wear out, especially in cells that are actively dividing, like stem cells. And also we have it in cancer cells too. The activity of telomerase in cancer cells is also high. And also activated memory T lymphocytes that have to divide and differentiate to give rise to T cells, specialized T cells, and more memory cells. But this does not even have chromosomes, no DNA, no chromosomes, so we should not be talking about telomeres here. And for neutrophils are specialized cells. Specialized cells like neutrophils have limited regeneration ability. They are limited in terms of cell division. They are not actively divided as specialized cells. Specialized cells lose their ability to divide, so the activity here too is not going to be high. So the cell that will transcribe a high concentration of telomerase will just be the activated memory T lymphocyte that have to divide many times. So which statements are correct? All polypeptides are coded for by genes. That's correct. There are genes in the nucleus coding for different polypeptides that make up proteins. All genes are made up of a sequence of nucleotides. That's correct too. DNA nucleotide that contains adenine, thymine, guanine, and cytosine. All gene mutation results in a change to the nucleotide sequence. Here, when there's a gene mutation, that's the definition of a gene mutation. There's a change in the structure of the gene, which is the nucleotide sequence, whether base substitution, base addition, or base deletion. But all gene mutations will result in an altered polypeptide. It's not correct. It's not all the time. That's what we have. That sometimes we have what is called silent mutation, where the new nucleotide sequence is still producing the same polypeptide because of the degeneracy of the genetic code. The generate one amino acid can be coded for by more than one DNA triplet. So it's possible that the newly introduced DNA triplet is still coding for the same amino acid. So it does not alter the polypeptide. So just 1, 2, and 3 only. A double-stranded DNA molecule was analyzed and 29% of the nucleotide bases were found to be adenine, and which percentage of the nucleotide bases will be cytosine? Of course, you remember the base pairing rule. The base pairing rule says A is bonded to T and G is bonded to C. So remember Shagaf data. Shagaf data, you have equal amount of A and T, roughly equal amount of A and T, as well as G and C. So the double-stranded DNA molecule was analyzed. 25% of nucleotide bases were found to be adenine. Which percentage of the nucleotide bases will be cytosine? So if 29 was adenine, 29% will also be thymine. And when you add these two together, it gives us 58%. So let's subtract 58% from 100%. Everything has to be 100. So 100, 58, that's 2. That's 42% left. So 42% to be shared between the both of them. And that will be 21% for each of them. So 21% for cytosine. So in some cells, non-coding sequences of RNA are removed after transcription. We call that RNA splicing. Remember, RNA splicing, the removal of introns, which are the non-coding sequences, and the joining of exons, which are the coding sequences that contain genes. So which row correctly states the name of these non-coding sequences of RNA and the type of cell in which non-coding sequences are removed? Of course, I just said name of non-coding sequence will be introns, not exons. So we are between C and D now. The type of cell in which we find introns, we don't find introns in prokaryotic cells, in bacterial cells. You only find exons in their operons, right? No introns, but we find introns and exons in eukaryotic cells. So the answer to this question is option C. We'll go to the next question, number 26. Water potentials were measured inside the leaf cell in the air spaces of the leaf and in the atmosphere. Now, which values of water potential would allow water to move from the leaf to the atmosphere? You will agree with me that for water to be able to move from the leaf, about the water potential, from these leaf cells, whether the palisade or spongy, into the, just a rough sketch to quickly go through this. So the air outside has to be, for the rate of transpiration, that's what we are talking about here, for the rate of transpiration to be high, for transpiration to occur, the humidity, which is the amount of water vapor, humidity has to be low. So water vapor here has to be, water potential here has to be very negative. Very negative. It has to be very negative, like minus 100. Then when you compare with the water potential here, this one has to be higher. So less negative compared to what you have outside. Less negative. Then inside the leaf, it has to be higher too. The least negative. So if you look at it, inside the leaf, it should be the least negative compared to the hair species here. Then outside the leaf should be the most negative, which is the smallest. Most negative means it has the least water potential, the smallest water potential. So that's the answer. So we move to
a lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Ajiro Tech do have a private tuition. So by my right, you can see that we have for the Edexcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, I think all you have to do is send an email to the sales team at ajirotech@gmail.com, and they would reply to you, talk about your time zone, talk about, and the fee is really quite affordable. Really, it's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable, and you get the very best of service. We also have for the E-levels as well, and also for the Edexcel. So we have for the Cambridge E-levels and Edexcel E-levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E-level, mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time. Even physics, for E-level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid, okay, and send an email to the sales team at ajiro@gmail.com, and they'll reply to you.
the next one, the next question, 27. 3 samples of liquid were taken from different locations in a plant and tested to measure the concentration of 4 solutes. Okay, the results are shown. We have concentration of solutes in mole per meter cube. Okay, so we have sample 1, 2, 3. This one contains sucrose. Remember, sucrose is the form in which sugar is transported in phloem. recall knowledge, that's good. Also, in addition, we have amino acids transported in phloem. We call them assimilates. The compound synthesized by living organisms like plant, assimilate, the organic compound. However, we know that xylem transport water and mineral ions, but however, too, we can have some mineral ions pass into or move into from xylem, move into phloem. And so it's not strange to have mineral ions like nitrate ions and magnesium ions in phloem. So the one that actually shows us that it's truly phloem is where we have a higher concentration of the two major substances transported by phloem, which are amino acids and sucrose. So this shows us that 1 and 3 were taken from phloem sieve tube element. Coming to the question. So 1 and 3 only. A heated band was wrapped around the stem of a plant at a height of 1 millimeter above the ground. So we have the plant 1 millimeter above the ground. So this is the ground. So a heated band was wrapped around the stem. The heated band 1 millimeter above the ground. Beg your pardon, 1 meter, not 1 millimeter, 1 meter above the ground. And left for 10 minutes. Now, when a heated band is left for 10 minutes, remember it will be entering the plant from the stem. So how will heating a small section of the stem to 60 degrees Celsius? Remember, plants are living organisms too with enzymes inside their cells that are sensitive to temperature. So how do they affect the transport of phloem sap and xylem sap between the roots and the leaves? Remember, phloem is a living tissue. Xylem is non-living. This still contains enzymes. The phloem contains enzymes that are sensitive to temperature, so it will be affected. But xylem, being non-living, will not be affected. So transport can stop for phloem because it is living, it has enzymes that will facilitate and proteins in the membrane that allow transport to take place in it. The diagram shows the structure of an amphibian heart and the movement of blood through it. As you can see in the amphibian heart compared with the mammalian heart, you did the mammalian heart, you have in your coursework, showing us that this is ventricle and there are what? There are actually atria. The right atria, left atria. Then one ventricle. Obviously, as you can see here, there is no septum. Separating, like we have in the, I just seen a valve. We're seeing valves for no septum. Okay, I'm trying to look in advance. So no septum. So which statement about differences between amphibian heart and the human heart are correct? The hearts have a different number of valves. Comparing the amphibian heart with the human heart, do they have a different number of ventricles? That's correct. The human heart has 2 ventricles. This has only 1. The heart have a different number of atrioventricular valves. Remember atrioventricular valves between the ventricles and the atria. There are 2 in mammalian heart or the human heart. There are also 2 here. So they have a different number of atrioventricular valves. That's not correct. They both have 2. A septum is absent in the amphibian heart. That's true. Present in the human heart. That's correct. So we have 1 and 3, one incorrect. A mutation in one of the genes that code for hemoglobin changes the affinity of hemoglobin for oxygen. So hemoglobin saturation, oxygen saturation, and here we have the partial pressure of oxygen. Now this mutation lowers the partial pressure of oxygen at which hemoglobin is 50% saturated. So let's say we had this as a normal curve. So we are saying that at, say, this is 100, this is 50. So at 50% saturation here, we have a particular partial pressure of oxygen. Now, we are now told that it lowers the partial pressure of oxygen at which, so it lowers it, makes it smaller, partial pressure of oxygen, at which hemoglobin is 50% saturated. So if you check this out, what it means is we have 50% saturation of hemoglobin. The curve is going to change definitely. So the curve is going to move like that. So such that it lowers it. To lower it, the curve has to move to the left. So if we check 50% saturation with lower, can you see? It is still 50% saturation, but with lower what? Partial pressure of oxygen. Partial pressure of oxygen has reduced. With lower. So that means that the protein is 50% saturated at a lower partial pressure of oxygen. Similar to Km. Just a small amount of oxygen is making it 50% saturated. Affinity has increased. So the effect of the mutation on the affinity of hemoglobin for oxygen is that it has become higher. And the change in the position of the oxygen dissociation curve for hemoglobin is to the left. It shifts to the left. This is left. Yeah, this is left. This is to the right. It's to the right like this. Shift to the left. So it's to the left of the main curve. So higher and shift to the left. Which processes are responsible for Bohr shift? Bohr shift is brought about by high concentration of CO2 and low pH. High concentration of CO2 and low pH. Of course, CO2 is acidic. So the moment CO2 gets into the blood, what does it do? It lowers the pH. It lowers the pH. It combines, it moves into the cytoplasm of a red blood cell, combines with water to form carbonic acid. And carbonic acid breaks down into hydrogen carbonate ion and hydrogen ion. Now this goes to hemoglobin. Hemoglobin picks this up to form hemoglobinic acid, making hemoglobin to release its oxygen. Right, so making hemoglobin to release its oxygen. Also, we have some CO2 binding to the terminal, to the amine group of terminal amino acids in hemoglobin. So CO2 can bind to the amine group of terminal carbamate, right? That's what it's called the chemistry. So here, this also reduces the affinity of hemoglobin for oxygen. Still Bohr's effect. So carbon dioxide reacts with hemoglobin to form carbamino hemoglobin. It's still part of Bohr's effect or Bohr shift. Carbon dioxide reacts with water to form carbonic acid. You can see this is leading eventually to the formation of hydrogen ions that we make hemoglobin to release oxygen. Still part of it. So Bohr shift is saying that high concentration of CO2 and low pH reduce the affinity of hemoglobin for oxygen. And you see, as a result of CO2 carbon dioxide reacting with water to form hydrogen carbonate and hydrogen ion, the hydrogen ion comes from this process, and it reduces the affinity of hemoglobin for oxygen. So it's part of it. Hemoglobinic acid is formed from the dissociation of carbonic acid. You can see the formation of hemoglobinic acid makes hemoglobin unavailable to bind oxygen to. So the three processes are responsible for Bohr's law. A student viewed a drop of human blood with a microscope and described a cell as having a very large diameter. A very large diameter. A very large diameter. Even though a monocyte is going to become a bigger cell, right? But it's very large as well. And if you look at it too, it's very large and it has a U-shaped, not like a neutrophil, but it's actually U-shaped, truly U-shaped. That of the neutrophil is lobed. You say it's lobed, right? This is U-shaped monocyte. So how many times must an oxygen molecule pass through a cell surface membrane to get from the air in the alveolus to hemoglobin in the red blood cell? So we can visualize this question like this, that we have the alveoli wall. And what do we have in the alveoli wall? We have the epithelial cells. Then we have an oxygen molecule inside the alveoli, the alveolus. And we have very close to it, we have the capillary. So here we are looking at the endothelium of the capillary. This is the endothelial cells that line the wall of the capillary. So the oxygen molecule is meant to move, can diffuse through this membrane of the epithelial cell. So this epithelial cell, this is endothelial cell of the capillary, epithelial cells lining the wall of the. So it moves through the first membrane, that is one, gets into the cytoplasm, moves through the second membrane, that's two, gets into the intercellular space, passes through the third membrane, gets into the cytoplasm, passes through the fourth membrane. Now it's coming into a red blood cell, wants to bind to the hemoglobin in a red blood cell. So it's going to pass through the membrane of the red blood cell. That's the fifth membrane is crossing, cell surface membrane. So 1, 2, 3, 4, 5. So the oxygen molecule is crossing 5. We'll have to cross 5. Asthma can affect children and adults. The air passages become narrower. The air passages become narrower, severely restricting the flow of air into the lungs. But we have salbutamol, which is a drug that widens the air passages to relieve the symptoms of asthma. How does this drug, salbutamol, relieve the symptoms? Widen the air passage. What widens the trachea, the air passage, the bronchus, the bronchioles, that make up the air passage? The action of the muscle. the smooth muscle there. The smooth muscle contracts, and when the smooth muscle contracts, it's going to narrow the air passages. When the smooth muscle relaxes, it widens the air passage. So the drug relieves the symptom of asthma by relaxing the smooth muscles. So it acts on the smooth muscles in the bronchi and the bronchial wall, making them relax. And when you look at this photomicrograph showing us the transverse section of the part of the trachea, remember in the trachea we have smooth muscles, we have cartilage, C-shaped cartilage. The smooth muscles can contract and relax to adjust the diameter. What we just looked at, here we have which letter represents the smooth muscle. That's what we are asking directly. When you look at this place, so we'll move on to the next one. Which statement about an infectious disease may be correct? An infectious disease is caused by a pathogen and transmitted from an infected person to a non-infected person. And usually we have some infectious diseases that are carried by vectors, like malaria, like sleeping sickness, carried by insect vectors. So we can actually have infectious diseases carried by vectors or passed from the host to a non-infected person. So let's go on. Says it can be caused by a prototist, right? Yeah, like I just mentioned, malaria. Malaria is caused by a prototist. So it can be caused by prototist. This goes. It can be transmitted by an insect vector. Transmitted, not caused. Yes, that's correct. If this had been caused by an insect vector, that would have been wrong. But transmitted as a vector, yes, it can be transmitted from mother to child. It's possible. Viruses, bacteria can pass through the placenta from mother to child. The same way nutrients, oxygen, and the likes can pass through the placenta. We can also have, for instance, HIV can pass through the placenta from mother to child. So the three, correct.
A lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Ajiro Tech do have a private tuition. So by my right, you can see that we have for the Edexcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, I think all you have to do is send an email to the sales team at ajirotech@gmail.com, and they would reply to you, talk about your time zone, talk about and the fee is really quite affordable. It's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable and you get the very best of service. We also have for the E-levels as well, and also for the Edexcel. So we have for the Cambridge E-levels and Edexcel E-levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E-level, mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time, even physics, for E-level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid okay, and send an email to the sales team at agile@gmail.com, and they'll reply to you.
Which statement explain why cholera has not been eradicated by vaccination? To eradicate a disease by vaccination, one organization that actually plays a major role is the WHO. And the WHO would supply vaccines. And you agree with me that you commonly have vaccines like polio vaccines around. You have the BCG for rubella, for German measles and tetanus. You even have the yellow fever vaccine. Yeah, right. And so we see quite a number of vaccines, but we are not so familiar with cholera vaccines. Cholera vaccines is usually given on special occasions when people want to travel to a place where cholera is endemic. And so because they don't have any prior, they don't have any previous immunity, they don't have immunity against it. They have not been infected before. So it's good they get this vaccine before going to such area where cholera is endemic. One of the reasons why we don't see this vaccine around is because it's not affordable. Limited availability of affordable vaccines. It has to do with affordability. And we are looking at why it is not eradicated. Protection takes several weeks to develop after vaccination. It's possible. Let's keep this. Now, the underreporting of cholera in the community, and WHO needs active surveillance. Feed them with information from active surveillance to be able to eradicate the disease. For instance, the WHO has been able to eradicate smallpox by vaccination because there were available vaccines, no limited availability of vaccines. And then there was active surveillance, reporting cases to the WHO. WHO was acting swiftly or acted swiftly to take care of those cases to eradicate. So you see these two very important for eradication of the disease. When we look again, we can also consider the fact that the reason why vaccination has not successfully eradicated it will be that when some people even eventually get the vaccine, what is the effectiveness of the vaccine? Protection takes several weeks to develop after vaccination, which means one could have been sick of cholera and, of course, transmitted the pathogen. Immunity to cholera decreases after 2 years. This could also be a factor why it has not been eradicated. Antibiotic resistance is a serious threat to global health. What gives rise to antibiotic resistance? Not completing your dose, unnecessary use of antibiotics too, and not completing your dose of antibiotic. Now, when you look at the first option, so it's a statement describes a step that can be taken to reduce the impact of antibiotic resistance. That means people should complete their dose. Antibiotics should not be used unnecessarily, only when prescribed. And one should not, just use antibiotics every time he has a disease. They're using antibiotics for a viral infection that can lead to or bring about antibiotic resistance. Viral infections should be taken care of by antiviral drugs. So what step can be taken to reduce a step that can be taken to reduce the impact of antibiotic resistance? We said antibiotics prescribed by a doctor should be shared. No, should not be shared with family members. No, this is unnecessary use of, to lead to unnecessary use of antibiotics. Antibiotics should not be used to treat, should only be used rather to treat infectious disease caused by viruses. No, this is against it. Just like I said, antivirals should take care of viral infections. Farmers should give antibiotics to healthy animals to prevent infections. This is a major cause of antibiotic resistance. Farmers giving antibiotics unnecessarily to their animals. The use of antibiotics by farmers. Vaccination programs should be used to reduce the spread of bacterial diseases, yes, so that there will be less need for antibiotics and there will be less cases of resistance. Tetanus is an infectious disease caused by a type of bacterium, and this bacterium produces protein that is a toxin which causes illness. Scientists have produced a vaccine for tetanus which contains a harmless form of the toxin called a toxoid. The toxoid is produced by mixing the toxin with the chemical formaldehyde, and this chemical binds to the toxin, making it harmless. My statement about the tetanus vaccine is correct. After vaccination, the toxoid in the vaccine will be ingested by neutrophils and displayed in their cell surface membrane. Of course, the tetanus vaccine containing toxoid does not contain antigen. This statement is wrong, outrightly. When formaldehyde binds to the toxin, it causes the toxin to completely change shape. This is also outrightly wrong. Now, we can consider this or this. But when we consider the latter part of this, the toxoid protein in the vaccine stimulates the production of antibodies, yes, which remain in the blood to provide long-term antibodies can eventually be destroyed. They don't stay. What actually gives long-term immunity are memory cells. Antibody get to be removed from the blood after a while. But memory cells remain in the blood for long-term immunity. Neutrophils like macrophages can also act as APCs, displaying the toxoid on their cell surface membrane to stimulate other B cells to carry out the immune response. So that's A. Thank you. What is an effect of the immune system of a reduced T helper cells? T helper cells produce cytokines that stimulate both the specific and non-specific immune system, that is both the phagocyte and the lymphocyte. So when you have a reduced number of T helper cells, like we have in the case of HIV/AIDS, HIV attacks T helper cells. So we are going to have a decrease in the destruction of infected body cells by T helper cells. T helper cells do not destroy infected body cells. T killer cells do that. T killer cells, the phagocyte. A decrease in the activation of B lymphocyte. B lymphocyte, like I just mentioned, cytokines can activate both specific and non-specific immune system. B cells or B lymphocytes are part of the specific immune system. They can be stimulated by the cytokines. They can be stimulated by cytokines produced by T helper cells. So when you have a reduced number of T helper cells, there's going to be a decrease in the activation of B lymphocytes. Is there going to be an increase in the activation of T killer cells? No, there's going to be a decrease in the activation of T killer cells too, because T killer cells are also activated by cytokines. An increase in the production of plasma cells, this is wrong. This is negating what has been said here. There's going to be a decrease in the production of plasma cells. Plasma cells come from the differentiation of B lymphocytes. So B lymphocytes differentiate into plasma cells, which produce antibodies. This brings us to the end of the correction. Thank you.